Hi, I'm Charlie Silver. I'm the CEO of MissionBio. We are a single-cell genomics company based in South San Francisco, and our mission is to help our partners leverage single-cell biology to accelerate the discovery, development, and delivery of advanced therapeutics to help get drugs to patients. We provide the only single-cell multiomics platform that offers true multiomics with the ability for both a genotypic and phenotypic readout of every single cell. So it does both DNA and protein simultaneously on every single cell uniquely. We offer it as an instrument consumable set. Down there is a picture of the Tapestri instrument. We provide one instrument along with kits that run different applications based on the content for that specific application. We also offer the ability to customize the content so our customers run any number of different applications on the platform. And we provide a pipeline and an analytics solution that's fully integrated at the back end to get our partners through to the insights that they need. The technology is, again, uniquely a single-cell DNA platform. Many of you might be familiar with other expression platforms. We are the only platform that provides DNA with a chemistry that's really good at extracting inhibitors from the nucleus that might otherwise inhibit the reaction, and then run chemistry to enable single-cell SNPs or indels or copy number at the gene level through to copy number across full chromosome level. So it's the only platform that offers every resolution needed for single-cell DNA. Sample loads onto the platform as a suspension. For solid tissues, it could come in as disaggregated cells, and we offer protocols for disaggregation as well as disaggregated nuclei. That suspension loads into the platform, which then individually barcodes up to 10,000 single cells per sample. And those codes are integrated along with an amplicon panel, which defines the content for any specific application. So comes off the platform, goes on to a standard Illumina NGS workflow with tens of thousands of single cells barcoded and amplified with the content of interest. Coming off the sequencer is typically millions of sequence reads that now have single cell accuracy defined by those barcodes. And then the visualization converts those reads into single cell clonotypes, again, defined by the amplicon content for the platform, which then gives exquisite resolution for single cell, which is typically lost in a bulk setting. So in contrast to bulk, we can now get information on combinations of mutations in every single cell, which would otherwise be lost with conventional sequencing. We provide catalog content for a number of different solid tissue indications, as well as some blood cancers. And we also offer the ability to customize with any amplicon content that can be run on the platform. We serve a number of different translational applications across different market segments as the leader in single-cell multiomics analysis. We serve translational cancer centers with the ability to resolve clonal heterogeneity for large-scale clinical research. We serve our biopharma partners with enhanced biomarker development, which provides really good sensitivity for response, resistance, and relapse in reaction to therapeutics. And we also support cell and gene therapy, where we enable accelerated approval and expanded product labeling. We also support the CRISPR genome engineering space with a tool set that's fundamentally used as a proofreader or a spell checker for CRISPR as a way of identifying what edits you've made. We've been a part of the NIST Gene Editing Consortium as a member for the last couple of years. We're the only single-cell DNA technology providing proofreading capability. And we support that space with the ability to get down to the ground truth of what edits we've made into the cell population. So rather than having to infer back from bulk, you get a true measurement. And we provide this really as a way of the space to standardize across different editing systems or different ways of editing the genome, different genome engineering systems, By standardizing the result, we provide a means for the entire space to be able to really standardize on their measurements. The cell and gene therapy development pathway has a number of different challenges because these therapies are extremely advanced and they're very complex. Relative to small molecules or antibody development, the design, development, manufacture, and deployment of these therapies is much more complicated and carries a number of challenges that we're all familiar with, from drug substance release assays through to chemistry, manufacturing, and controls, the CMC testing that validates and performs the release testing on lots, through to drug product stability testing, patient monitoring and follow-up once the drugs are in the patient. There are challenges with understanding what the drug has done. Many challenges across the cell and gene therapy space. And these challenges are really well served with single cell analysis. Single cell provides the means for a high throughput assessment that combines transduction efficiency and vector copy number for improved specificity, and importantly, for reduced variability of all of the parameters that go into characterizing the drug product for development or for release testing. It provides the ability for both on-target and off-target assessment for gene editing, including translocations, as well as vector copy number, with the ability to look at single-fold changes in copy number to understand how the product has been integrated across the cell population. And finally, the sensitive immunophenotypic capability of the platform is tremendously useful, both for characterizing the product and for biomarkers of how the product deploys in patients. And the ability to measure vector copy number on every single cell across the population, and on that same cell to be able to measure the functional result of different vector copy number changes, provides really unique capability with a very robust characterization set. We support customers around the world. We are adopted by more than half of the cancer centers here in the US, and more every quarter, as well as many worldwide. And we also support more than a couple dozen different pharma programs, both across biomarkers and for cell and gene therapy and genome engineered products. I'll talk about biomarkers for a couple of minutes as an example of how the platform is deployed for drug development pipelines. And for biomarkers, the resolution afforded by single cell is really used to accelerate drug development pipelines. At the front end of a pipeline, to help identify the attributes and composition of a product, which we all know increasingly is very important to define at the front end to really ensure the success of these products as they move through testing and through the FDA package. Platform is also used by drug companies to monitor patients while they're on treatment for really exquisite biomarkers of response and resistance. And finally, it's used really across the board for reverse translation. In a setting where there are lots and lots of retrospective patient samples, whether it's a trial that's been run recently or years behind, it's used as a way of looking across those samples and understanding where's the signal that can be deployed for the next generation of therapeutics. And that's a very common use case for a lot of our biopharma customers in biomarker development. I'll show an example of how the platform is used for biomarkers. And this is an example where single cell really elucidates the complex clonal architecture of co-occurring mutations and helps understand how that clonal architecture evolves in response to therapy. As an early readout of how drugs will work in patients or how to design the right trials so they will work for the patients that they're targeting. This was an example. This was a study that was run by Agios in conjunction with a number of cancer centers across the US, where they took a look at a cohort of patients who were treated with an IDH1 inhibitor and then developed resistance into an IDH2 pathway. And they used the platform to understand those resistance mechanisms as a way of understanding how they can deploy combination therapies or redesign a trial in a way that would be more effective for these patients. And what you're looking at here are what we call fish plots. So these are plots that longitudinally monitor patients, where there's a baseline for every patient. And then you can see samples during treatment that show how different mutations come and go. And what you're seeing here are three different fish plots from patients that are representative of different mechanisms of resistance. The one on the right shows a patient that presented with two different baseline clones of IDH1, a dark blue and a light blue. You can see that the dark blue was cleared, the light blue expanded and evolved into an IDH2 clone, which was predominant at relapse. Panel in the middle shows an example of a patient with very complex clonal architecture at baseline, where an IDH1 mutation through treatment actually went away and switched isoforms into the IDH2 mutation. Very interesting resistance mechanism, where the complex clonal architecture actually evolved into isoform switching through to relapse. And then the panel on the right is an example of a patient which had IDH1 at baseline in addition to IDH2. And this one is really interesting because that patient had IDH2 at baseline, which was not detected by bulk sequencing. It was uniquely identified by single cell. And that's an example of how single cell DNA can be used for the exquisite sensitivity of barcoding, because it is barcode sequencing, to be able to get sensitivity orders of magnitude below the typical 5% cutoff that you would get from bulk sequencing. And Tapestri can provide much better sensitivity than that. So all of these are examples of how the platform can show the mechanism of resistance, specifically in this case to IDH1 therapy. And it showed how the mechanism of resistance is complex, it's polyclonal, and ultimately out of this study, it shows support for the use of combination strategies or sequential treatment strategies for these patient cohorts. Tapestri is also used for the cell and gene therapy partners to help them develop and characterize their drugs. And it's used as a way of integrating single cell analysis into current and existing gene therapy development and production workflows as a drop-in replacement for conventional technologies. High throughput single cell assessment can combine transduction efficiency and copy number, which are both very important assays for characterizing these products, in a way that improves specificity and reduces the variability for a very robust measurement, while reducing the time to answer. Single cell can provide both on-target and off-target gene editing information, including translocations to understand the heterogeneity across the cell population of the cell product. And finally, the ability to measure both DNA and protein together with vector copy number and all of the other parameters that you can get out of a Tapestri assay. That fully integrated capability provides a really robust data set for all of the parameters that are needed ultimately to be able to assess dose and toxicity for these products. Ultimately, the time to answer is what's most important, because conventional assays require lengthy cell outgrowth, which can take which can take weeks up to months of time to be able to characterize just a single dose of the product. Single cell obviates the need for lengthy cell outgrowth, so you can sample the product directly and make a measurement within days as opposed to weeks or months. And that time to answer is absolutely crucial in getting these drugs to patients on a time scale that they need it. It's used to optimize integration conditions, whether it's a lentiviral vector or an AAV vector. It's used to optimize the conditions and to measure the progress toward desired results. And it's used both in development to be able to optimize the system on a much faster time cycle, and in production to streamline the testing and release of manufactured cell products. Here's an example of data that was generated for CRISPR editing. through adjuvant technologies. And you can see basically every combination of what's needed to characterize CRISPR systems on each of these panels. On the upper left, this was a 3-gene editing system for HBB, CTLA, and RAB11A. So 3 genes were edited in, and the platform was used to measure on target for each of these genes, as well as putative off target. And the panel on the upper left shows the different combinations across the cell population. The upper right shows different zygosity states, and this becomes very important, especially when we're manufacturing a therapeutic that has improved efficacy for homozygous edits versus heterozygotes. It's really important to understand how you've edited, whether it's one or both alleles. And the panel on the upper left shows that you can get every single combination for these different mutations that were edited in. Blue shows before optimization, red shows after we've taken a turn of optimization on the guide RNAs, and you can see that you get a very different answer. And for these multiple edits, it's important to measure that at the single cell level because you could never get that information in bulk. The panel on the lower left shows an example of a translocation, and then it all rolls up together to the panel on the lower right, which shows every combination of possible translocations between on target and off target, and every combination of on target and off target. So it's really a very robust and rich characterization mechanism as an assay for characterizing these genetically engineered cell products. The assay is also used to quantify cell products without complicated clonal outgrowth as a way of directly measuring the product with really just exquisite linearity and precision, which provides a very robust and repeatable assay across different samples. And you can see that here, the panel on the left shows actually 25 different samples in serial dilutions. So there are 5 different dilutions. Blue shows vector positive, red shows vector negative. And you can see a very sensitive measurement, where on the right, you see great linearity, which is afforded by this barcode-based sequencing approach, where, with 5 replicates across the 5 different dilutions, you get just exquisite linearity, which is what's really essential for these assays. We provide services for the cell and gene therapy space. So we provide this as an internal service offering so we can bring our partners up and running very quickly with a custom built and turnkey assay that's specific for their pipeline or specific for the specific drug of interest. We run this as an internal development program in partnership with our customers. And we can then transfer that over to a CRO or CDMO of our customer's choice. We work with our partners to design a specific study, which we then build and validate and then turn into a turnkey assay, typically with turnkey and custom analytics that are customized for whatever assay our partner needs. And the deliverable is then the data report and the visualization that gets them through to all of the insights they need for characterizing their drug product. And from there, we can either continue to scale it up internally, or we can transfer to them or to a CDMO of their choice. For information on how to partner with us as a company, my contact information is here. Matt Cato is our head of cell and gene therapy. He's also at this conference, and his contact is shown below as well. There have been tremendous advances in targeted therapies and cell and gene therapies, which have just tremendous promise and impact in improving the outcome and welfare of patients. And single cell biomarkers enabled by Tapestri, as well as a full suite of characterization assays with a reduced time to answer, really supports the space by enabling drugs to get to market and to get to the patients who need them faster. Thank you.